Today we're going to be working on Chapter 12, talking about different drugs for different microbes that we have and the effects on the host. And first of all, we want to look at the goal of antimicrobial chemotherapy. This is where you administer a drug to an infected person and it destroys the infective agent without harming the host cells. And we can get these antimicrobial drugs either naturally or synthetically. But we need to remember the perfect drug doesn't exist, but we have to balance the characteristics of one drug against another and come up with a satisfactory compromise, hopefully. Table 12.1 lists the characteristics of an ideal antimicrobial drug that you'll need to look at in your textbook. Now, antibiotics are what we use a lot of the times. And antibiotics are metabolic products of aerobic bacteria and fungi. And they can inhibit or destroy other microbes. For example, bacteria in the genera Streptomyces and Bacillus, and molds in the genera Penicillium and Cephalosporium. And so by inhibiting the other microbes in that habitat, these ones that produce the antibiotics have less competition for nutrients and space. And this is how Alexander Fleming discovered penicillin accidentally with the mold that was on food, that it came from there. Now, this table shows the selected microbial sources of antibiotics. You see mold is penicillin and cephalosporium, and then you've got bacteria that will produce gentamicin and bacitracin, and you've got some filamentous bacteria that produce the streptomycin and erythromycin and the tetracycline and vancomycin. You may have be familiar with some of these. This is the terminology of chemotherapy. If it's a chemotherapeutic drug, it's a chemical that's used to treat or relieve or keep the disease from coming back. Prophylaxis is when you give a drug to prevent a possible infection if the person is at risk. Antimicrobials, that's a term for any antimicrobial drug. Antibiotics, those are the ones that are produced by metabolic processes of microorganisms. And then you have the narrow spectrum, which they work against a very limited group of microbial types. It's very effective on mainly gram-positive bacteria, and the broad spectrum affects a wide variety of microbial types, and it's very effective for both gram-positive and gram-negative. Now, sometimes antimicrobial drugs can be selectively toxic, where they kill or inhibit the microbial cells, but they don't damage the host tissues. And sometimes the characteristics of the infectious agent are very similar to the vertebrate host cell, and so it's hard for them to be completely selectively toxic because they're going to have more side effects seen and so forth. There are 5 major components that are useful targets in an actively dividing cell of these antimicrobials. First is to inhibit the cell wall synthesis, then to break down the cell membrane, either the structure and or the function, to interfere with the nucleic acids DNA and RNA and inhibit protein synthesis, and to block key metabolic pathways like folic acid metabolism. So these are the major targets. It would behoove you to look at this closer and see which drugs do what. For cell wall inhibitors, cell membrane, nucleic acids, protein synthesis, or the metabolic pathways. Now, if we look at the spectrum of the antimicrobial, we've already mentioned, narrow spectrum, that's just a small range of microbes. They target a specific cell component that's only found in certain microbes. Bacitracin is one. If they're medium or broad spectrum, they have the greatest range of activity, and they target cell parts common to most pathogens like ampicillin and tetracycline. So this leads us to our first concepts question. A drug that targets lipopolysaccharides in a gram-negative cell would be considered, is it specific or is it over a large group? Selectively toxic.